Hi everyone, our chosen topic is using CRISPR gene editing tool as a way to treat genetic diseases in animals for the first time. And this presentation is brought to you by Matthew Cook, Nathan Cheng, and Ishar Shah. CRISPRs, or clustered regularly interspaced short palindromic repeats, are repeating DNA sequences in the genomes of prokaryotes like bacteria and archaea. They were discovered by Yoshizumi Ishono in 1987, a Japanese scientist who was analyzing a gene and accidentally found a series of repeated sequences. At the time, there was insufficient amount of DNA sequence data and was therefore disregarded. Come 1993, and a team of researchers led by J. D. Van Ebden found that different strands of Mycobacterium tuberculosis had different spatial sequences between their DNA repeats. In 2002, researchers Francisco Mojica and Bud Johnson named these different spatial sequences as CRISPRs. From when CRISPRs were first discovered, they were highly disregarded and thought to be an insignificant DNA repair method present in the thermophilic archaea and bacteria. In 2005, however, scientist Mojica and his team found that spatial sequences found in prokaryotes were similar to the sequences found in bacteriophages, viruses, and plasmids. They found that viruses could not infect spatial sequences with homologous bacteria in them, indicating that these sequences play a role in the immune system of pure prokaryotes and functioned into a defense mechanism to prevent repeated infections from the same virus. Following Mojica's discovery of CRISPR's function as defense mechanism, Markova and her colleagues conducted an analysis of CRISPR and Cas genes as they believed that CRISPR-Cas systems' functions were similar to RNA interference, where the protein silences genes by splitting mRNA. George Church and his team conducted an investigation using the CRISPR-Cas9 system and discovered that by modifying the RNA to target a specific part of the genome, the CRISPR-Cas9 system could be used as a genome editing tool. In recent years, it has already been used in certain areas, such as switching off genes that withhold the production of lipids in microalgae, allowing for higher production of lipids and biofilm. In fact, CRISPR-Cas9 could even work with curing genetic diseases like cystic fibrosis, and as there are already a wide range of applications for using it in the treatment of diseases which include cancer and infectious diseases. The use of CRISPR-Cas9 in the treatment of diseases and genome editing is still fairly new, and is still being modified to further improve its use, and results in targeting specific genes to mitigate risk and make it more successful. With this new method of genome editing, new avenues in medicine have opened up. It could possibly be the next biggest treatment method for genetic diseases.
Now that you have some background knowledge on the history of CRISPR, I'll be explaining the mechanism behind it as well as some current limitations. CRISPR is a gene that codes for an enzyme that acts as a pair of molecular scissors, making targeted incisions in the DNA. The acronym CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which compactly describes the nature of the gene. As mentioned, there are palindromic repeats of nucleotides which are surrounded by small sections called spacers. These spacers allow the protein coded by CRISPR to recognize where to cut the DNA. While the CRISPR system is a naturally occurring process found in bacteria, molecular biologists have modified it to accommodate animals and humans. This links back to the BABS1201 theme of thinking like a scientist, as only through trial, error, and collaboration have scientists developed this technology. There are two components of the system, the CRISPR-associated 9 protein, or Cas9 protein, and the single guide RNA, or sgRNA. The Cas9 protein is an enzyme that the CRISPR gene codes for, which, as mentioned, performs the actual gene editing. The sgRNA is an artificially made sequence of RNA that guides the Cas9 protein to the desired gene. This occurs as the sgRNA contains a sequence complementary to said gene. Now let's delve into the steps involved in this revolutionary gene editing technology, which is expected heavily revolves around the theme of genetics. First, a viral vector is used to transport the sgRNA and Cas9 protein into the targeted cells. Then the sgRNA binds to the Cas9 at 2 sites, allowing it to read where it needs to go. Upon reaching the desired gene, Cas9 then causes a double-stranded break of the DNA, removing the gene. At this point, the CRISPR system has completed its job, and it's now up to naturally occurring mechanisms to repair the cut. This can happen in two ways. Either there is a random insertion or deletion of nucleotides at the site, or the homologous copy of the DNA is used as a template for repair. Even though CRISPR is much more successful than previous gene editing technologies, it still has its limitations. The biggest issue currently faced stems from the fact that Cas9 is not 100% accurate with its edits. This results in what is known as off-target effects, where unwanted mutations arise, sometimes leading to the formation of tumors. Another problem is the viral vectors used to transport the CRISPR components, as they may initiate a heightened immune response.
Many developments and experiments have been made in the field of CRISPR, and the field looks to be extremely promising to this day. For example, the first use of CRISPR-Cas9 to correct a genetic disease was performed by Wu et al. In the article Correction of a Genetic Disease in a Mouse via the use of CRISPR-Cas9 in 2013. This article starts by prefacing that the genetic manipulation using the CRISPR-Cas9 system has been demonstrated extensively by this point in many different species, but never correcting a genetic disease in particular, and that's what they would be attempting in this instance. The target gene in this experiment is a 1BP deletion in the exon 3 of the CRIAC gene, leading to a stop codon being coded prematurely and causing the development of cataracts in mice, as seen in the picture on the top right. Designing 5 different sgRNAs, they performed preliminary research involving the use of stem cells, finding sgRNA4 to be the most effective. Wu et al. Then performed the main experiment, shown in the bottom right, injecting the Cas9 mRNA and the sgRNA4 into zygotes suffering from the genetic conditions. Out of the 135 blastocysts that ended up developing, which was 91% of the total zygotes, a total of 22 were born. Out of these 22, 10 were identified to have genetic modifications of the mutant allele, with 4 of them carrying the corrected allele. These 4 mice were free of cataracts, identified by the lack of the opaque lenses of the mutant mice. Furthermore, Wu et al. Go on to do another study supplying an exogenous to WT-stranded DNA oligo to see if efficiency was increased. In this experiment, a total of 29 were born from 178 blastocysts. Of these, 9 were free of cataracts. Finally, the fertility of the repaired mice were tested, with the offspring collected from 4 of them, all carrying the corrected gene. This study, the first of its kind, even though it had a rather low success rate, proved that the correction of a genetic disease in a mammal is possible. Wu et al. Identified a problem in the experiment was the potential for off-target mutations, mentioned before, which could have influenced the low birth rate of the mice. Identifying 10 potential off-target sites, they put through 12 corrected mice through DNA sequencing, finding no off-target sites in 10 of them, and the remaining 2 mutations were detected in 1 of the 10. Therefore, Wu et al. Proved that the genetic correction of genetic diseases in mammals were possible, even through the low success rate. Overall, since Mojica's discovery, many and continued developments in CRISPR continue to provide promise in the field of medicine. Many people around the world suffer from genetic diseases, and CRISPR has the potential to alleviate that.